Hello, my name is GeritPfuhl. This is a pre-recorded presentation for the 2020 meeting of the Society for Mathematical Psychology. And together with Håkon Kjelmeland, we present a novel descriptive model to understand how we make probabilistic inferences. The data was kindly provided by Steffen Morris, James Julien, and Rick Adams. And our work is supported by a grant from the Norwegian Research Council. Experimental data has shown that we can be insensitive to prior information leading to the well-known phenomenon of base rate neglect, or insensitive to the likelihood leading to the well-known phenomenon of conservatism. Such under or overreaction has been proposed as mechanisms for hallucinations and delusions, and these are hallmarks of schizophrenia and also known as positive symptoms. Since the 1960s, probabilistic inference has been studied in the lab with the earn or beats task. In this task, prior probability and likelihood are told, and new evidence, the beats drawn or observed. Here we will present a mathematical model that describes the decision process of estimating the posterior probability for each beat sequentially observed. Our model employs three parameters: a parameter resembling probability weighting, that is how accurately one can estimate the posterior probability, a parameter on the precision of estimating the posterior probability, and a parameter describing the number of new evidence considered in inferring the posterior probability. We will show that biases in belief updating, the systematic deviations from the true posterior probability estimate, depends on task features, and that patients with a diagnosis of schizophrenia have accurate posterior probability estimates, but those are based on fewer samples and often are less precise. Probabilistic inference should be done according to Bayes' theorem. However, in real life, there are rarely only two choices, and there are only two models or hypotheses of the world. So often, exact Bayesian inference is nearly always impossible. But we can explain experimental data gathered in the beats or earn task, like the insensitivity to the prior or a likelihood, by adding parameters to tweak the calculations. These are the descriptive models, and a range of models have been developed in the last 50 years. Though to our knowledge, most of them address the accuracy, but not the precision and how many data is considered. Though another hallmark of patients with schizophrenia is actually poor cognitive abilities, including less good working memory, which may actually spill over into how many beats they consider when making probabilistic inferences. So in general, the underreactions, the likelihood can be captured with an exponent a being smaller than 1. So let's illustrate that. Given a certain prior and likelihood, the posterior will be a more precise estimate, with the posterior mean being between the mean of the prior and the likelihood distribution. And it will be also more precise, so the variance is smaller. In case of a broader prior distribution, we find an underreaction, in the extreme case a base rate neglect, and the resulting posterior is shifted towards the likelihood. We can achieve this also by assuming that the likelihood has less variance, or our estimate of the likelihood is more precise. Then again, the resulting posterior is shifted towards the likelihood distribution. The reverse happens if our estimation of the likelihood is less precise. Then the resulting posterior will remain closer to the prior. So this is then conservatism. Again, we can also find conservatism if actually the prior probability is more precise. Now we can also find that both the prior and the likelihood are less precise, meaning that distribution are broader. And then also the resulting posterior probability estimation will be broader than I've shown in the first example. Such a broad distribution will still on average give accurate estimations, but these are not precise. So this is also illustrated on the right-hand second row B. So similarly, like you need to draw more from a broader distribution to get a more accurate estimate of the true value. On the other hand, a systematic bias is seen if there's good precision but deviates from the true value. This is illustrated in the first row. An ideal Bayesian observer should have neither a systematic bias, so being very accurate, nor being imprecise. So that is illustrated in the last row here. So the task employed since the 1960s asks participants to estimate whether a sequence of colored beads comes from jar A or jar B. The jars differ in the ratio of the colored beads, so which color of beads is in the majority. participants are told the proportions in the jars and the prior. In the clinical setting, in the data set that we analyzed, there are always 2 jars, so the prior is always 50%. So the proportion in the 2 jars can be either symmetric and very distinct, so very easy, so 80-20 or 20-80, or they can be more difficult to discern, like when one jar contains 60 blue beads and 40 red beads, and the other jar contains 40 blue beads and 60 red beads. Or they can be asymmetric, so one jar contains 50 blue and 50 red beads, and the other one has a majority of blue beads. So how does this then look like? So again, the participant is first told that there are these 2 jars, the proportions are explained to them, and often also there's a control question without any bead first, there's no bead having yet drawn, ask what are the chances, and the correct answer is there 50%. Then here we see the first bead drawn is actually red, and it's asked to rate whether it's mostly coming from a jar with the majority being red beads or from the jar with the majority being blue beads. Then again, a rating is requested after seeing a blue bead, then again after red bead, and again after red bead. And that goes on for 10 beads. The rating is here illustrated on a 7-point scale, but also 11-point scale or continuous scales are used. So we can model this decision-making process with 3 parameters. So we have a parameter that captures the systematic deviation. So if the systematic deviation, as the parameter A is smaller than 1, we actually see conservatism, whereas if the parameter A is larger than 1, we actually see, in the extreme case, when A would be going to infinity, base rate neglect. For A equals 1, we actually find that the subjective posterior probability estimate is the same as the objective or true posterior probability. So there would be no systematic deviation. There's no bias. Then we also have a parameter for the unsystematic deviation from Bayesian probabilities. And that could indicate belief instability or or the precision, as I illustrated before. And then we also have the number of bits considered. We gave it the abbreviation, the letter M, so it's not memory as in cognition. And if one has an M of equal 1, one only considers the last bit. Now, we model data from 193 healthy participants and 148 patients with a diagnosis of schizophrenia. And here we see an example of 2 participants that have an accurate estimation of the posterior probabilities. So in both cases, we see that A equals 1, but they vary in the number of beats considered. Here only 1, whereas here all 10, and then the precision. So this participant is considerably precise. This participant is not very precise. And here we see how the rating actually in the 80-20, 20-80 sequence. And here we are seeing it for the 60-40, 40-60 symmetric sequence. On the left-hand side, we see here very certainty by the participant after seeing the 3 red beads. 100% certain it comes from the jar with the majority red one, but then, and that is here captured with a memory, the memory parameter being 1, reversing its estimation from which beat the jar is coming. We see the superficially similar, but in detail a different pattern here, because again the same sequence, yeah, all participants receive the same sequence, but here starting with a minority judgment. That's why it's here captured in the larger sigma. These estimations of the parameters A, M, and sigma are based in this data set on 4 sequences. So these are just examples. So what do we find overall when we look over all 4 data sets? There we actually see that for the systematic deviation, that is here on the x-axis, and the ideal would be here the gray line 1, that the patients actually on average, also seen here on the box plots underneath, are a bit closer to 1, particularly in the symmetric sequences, whereas the healthy controls have a smaller value, and it's always also less than 1, so they are far more conservative. Though for the asymmetric sequences, we actually see pretty good estimation, very close to 1 for the patients, and far more diverse and far more kind of indicating some base rate neglect phenomenon in the healthy controls. What about the unsystematic deviation? Again, that's shown here on the x-axis. We find some really less precise estimation in patients for 60, 40, 40, 60 sequences. It's also the case for the 80, 20, 20, 80 example sequences, so easy ones, but it's not the case for the asymmetric sequences. Remember, that was the 50, 50, 80, 20. They're actually the patients, though they have, again, a large tails, it's a quite heterogeneous group, but on average, or the median as seen in the box plot, doesn't make them less precise than the healthy control group. Now, what about memory, or how many beats they consider? If you again look first at the asymmetric one, we see that the patient group most of the time also considers 10 out of 10 beats. It's nearly always the case for the control, for the healthy participants. So it's less the case actually in the symmetric sequences that are hard or difficult. There are up only 2 or 3 beats are considered by the patient group. Though there is a caveat here, the no data set contains all 3 types of sequences. So there might be differences in the exact details, how the tasks have been presented, also the instructions, as well as the patient groups can vary. So overall, probability weighting depends on contextual features. Here, the type of sequences. We find underweighting of data for symmetric sequences, but overweighting of data for asymmetric sequences. But overall, the systematic deviation was always larger in the control group. So the patients are actually better in estimating the true posterior probability. The number of beats considered is smaller than in the patient group. So, and the lower number of beats considered does not imply larger systematic or unsystematic deviation. So there was no correlation there. Participants with a diagnosis of schizophrenia are noisier. So they are on average less precise, but that does not make them less accurate. So that could be due to an imprecise representation of those distributions. And what should ideally vary the prior by using different number of equal jar types. So though overall, we do not see evidence for aberrant probabilistic inference in patients. Thank you.